Okay, good morning, everyone. Welcome to the last third of Bio 1A. 6 weeks from now, summer vacation begins. And trust me, everybody on campus looks forward to it, faculty and students. My name is Gary Firestone, and that's my email address. And my office hours are going to be Wednesday, 11 to 12, Thursday, 2 to 3, in 2084 VLSB. To tell you a little bit about myself, I'm just starting my 30th year on campus, actually, on the faculty. And my area of research is hormones and control of cancer cell growth by hormones and phytochemicals that work in hormone-like manner. And so let me get a sense of you guys. How many of you are intended MCB majors? Okay, IB majors, plant biology, other kinds of science, ESPN, okay. Any of you humanities majors that haven't been taking the class? What are you guys, English, history, or what are you guys? Linguistics. Oh, someone who can speak. Very good. We need people like that. Now, the most important question I can ask you guys, how many of you can still earn points on your NCAA brackets tonight? Yeah, okay. I was knocked out after about the second round. So one of my kids at 8th grade class did a smackdown with my lab on the NCAA brackets, and they're killing us. They're absolutely killing us. The first 3 people are 8th graders. So we are not smarter than 8th graders, but maybe 5th graders in terms of what we do. For my lectures, what you'll be responsible for is what I mentioned in class. Okay? The book has lots of extra stuff. It's fun to read. Anything extra in the book, you won't be tested on. You're only tested on what I do in class. Secondly, I have a reader available. Have many of you guys been able to get a hold of it? Okay, you'll see it's just a written down version of what I put on the board, a few extra things. There'll be, again, what I say in class is what's important for what you need to know. There'll be a few extra things in the reader, and I'll say a couple things on the board once in a while that's not in the reader, but you're responsible for what I do put in class, okay? So today, let me give you an overview first before I talk about today. The first 6 lectures are going to be cells and tissues, hormone action, human reproduction, and stem cells. The next 6 lectures are going to be development, and then I call a systems flyby, because you can't do physiology in 4 or 5 lectures. It's pretty hard. But I'd give you a little taste of digestive, circulatory, respiratory, excretory, immune, possibly nervous system. The common thread is going to be hormone action. How do you regulate these systems? Okay, so I think that it's something you can, we'll talk about as we go through it. And certainly for human reproduction, you have to understand how hormones work to have any understanding whatsoever of human reproduction. So I consider it an important part of understanding some of this. Okay, so today we're going to do cells and cell-cell interactions, cells and tissues, the 4 different tissue types, epithelial, connective, nervous, muscle. And then we'll do possibly organs. And then in the reader, there's questions on page 13 that will help you see if you understand what we talked about. Okay? And these are the reader pages as we go through it. And the suggested book readings, which is close to what was on the syllabus. Okay? Any questions on that? Okay, and as you'll see my style, I am not PowerPoint whatsoever. I'm into chalk. It's a great technology. You put it on the board, it works every time. Okay? I like that. Anyway, just my approach to it. I like to start out talking about the hierarchical organization of cells and tissues. Some of this is intuitively obvious, but I think it's a useful discussion point as an introduction to everything. And then as you've been talking a lot about molecules that are expressed in cells, there's an expression and a function. You have about 200 different cell types. That's a number you do not need to know for any reason. There's about 50 trillion cells in your body, but only about 200 different cell types. The cells themselves can come from 3 different sources. Again, this is some of this should be fairly obvious. You can have a cell division like in mitosis, which I know you guys have talked about. You can have cell fusion, such as the gametes, haploid gametes forming a zygote. And more recently, you can have a fusion of a cell with a nucleus, an enucleated cell with a nucleus, and you can get a viable cell. So this is more of a more current definition. And this is just stuff I know you've here and there. Now these cells, which will be a topic today, work together in functional units. You have cell-cell interactions, which is really the heart of what we'll be talking about today. And there's a functional coordination. And this functional coordination allows for tissue function. Because you want, for example, all the muscle cells in your heart to work in a coordinated manner. If they're all independent, you wouldn't bode very well for a heartbeat, right? If they all did their own thing. They have to work coordinately. And then these tissues come together and form organs and organ systems. And all this is interrelated. Now, the reason I bring this up is severalfold. One is when we talk about how hormones work, they come in, the hormones actually interact with specific cells, causes a response, changes the molecules in the cells, but also changes how the cells interact and form tissue or function in tissues and in organs. You also have the whole issue of stem cells, which are mostly undifferentiated, form differentiated cells. And the idea is, say you want to develop new nerve cells to counteract paralysis, for example. You want to put the stem cells, turn them into neuron cells. These neuron cells have to function by certain molecules that form. But in addition, these cells have to work together in tissues and in organs. It's not just creating a differentiated cell type. It's pulling the cells together to work in a tissue. Same is true with gene therapy. In gene therapy, the same thing. You're missing something genetically. You add the gene to the cell, and you figure that's all you need. Not necessarily. You need to have these cells work together in coordinated units. So sometimes you can express a gene. If you express it at the wrong time, or the wrong amount, or in the wrong context, you can express the gene in a cell, but it won't necessarily form a tissue. And that tissue won't necessarily function in the context of an organ. And so I bring this up because a lot of what you guys, I imagine a lot of you either going to be going into medical school, research, CEOs of companies, things of that sort. You're going to be dealing with this issue. You could have a company, for example, that is sure has a great gene therapy approach. But until you test it in the context of tissues and organs, you don't really know if it's going to work or not. And that's the challenge. That's what you'll see in the next X amount of years, is trying to figure out how to actually do that for real. This is the overlay to things I'm going to be talking about the entire time. So when we talk about how hormones work in a cell, it's really in the context of tissues and organs. We talk about how tissues and organs work, it's in the context of gene expression and how cells function. It's all interrelated and all very important to come together. Okay? So, that's the segue for cell-cell interactions. Here's a diagram of just some cells sitting in a tissue, a generic tissue. And these cells interact with each other. They can communicate. And you have various junctions that allow for contact and communication. And I'm going to first talk about the communication junctions, or gap junctions. This is for communication. Now, in gap junctions are basically pores in between the cells. I'll just draw it like this. Here's the cell membrane. So these pores allow the movement of small molecules in between cells. Virtually all cells have this kind of communication of pores, which allows small ions, small molecules such as amino acids, certain kinds of lipids can get through. Secondary messengers such as cyclic AMP is sometimes it's thought to go in between cells. Now, virtually all cells can do this, and virtually all tissues. Exceptions are cells that are free moving, free swimming. Spermatozoa, which is what we'll talk about next week. Red blood cells, we'll talk about the week after that. So those cells are exceptions. They do not have gap junctions. But all sorts of other cells have them, and it allows these guys to communicate with each other. Now these pores allow for synchronized function. An example is muscle contractions. For example, cardiac muscle during heart beating and uterine muscle during labor. So it's not a coincidence that these cells all contract together or beat together. It's because they're communicating these gap junctions that go through these pores that go through the cell. Now, what are gap junctions made of? One can view this as this ear thing is really not working here. These gap junctions are made of what's called connexins. And there's 6 of them. So if here's the pore, there's 6 of these guys come together, and these connexins is the protein. And these are transmembrane proteins. And each cell supplies 6 of the connexins to the corresponding cell. So they come together and they form a connexin, a full unit that creates a functioning pore in these cells. As you can imagine, you can have disruptions in communication. For example, a lot of cancer cells express wrong and incorrect amounts of connexins, and therefore the communication is all messed up. And so there's several disease states where that's the case. Okay? Questions on gap junctions. They're pores. Fairly nonspecific, more based on size than anything. Question in the back. Yeah, the question is, do you produce more connexins than actually form a functioning gap junction? Is that more or less your question? The answer is yes. But you need 6 to come together to then form the functioning unit. If there's others hanging out in the membrane, it doesn't appear to affect it too much, as long as you have enough of the other complementary connexins that come together. The other thing, they have to be the same type of connexin. One of the very common ones in uterine muscle and so forth is connexin 43. Well, if one cell is connexin 43 and the other one has a different, that's a molecular weight size, 43,000. If you have like connexin 37, it won't work. It has to be 43 with 43s. So there's a set of rules on how they come together. And, but it's very important in terms of communication. Other questions? Very good question. Yes, another one in the back. Yeah, a very good question. The question is, can you have different sets of connexins in cells and therefore prevent communication when you don't want communication? The answer is yes. That occurs not so much in the same cell type, but in the boundary of different tissues, for example. That you'll see that sometimes. Absolutely. You can also have cells, and for your third question you were going to ask, I'm sure, can cells express 2 different kinds of connexins? The answer is yes. Okay? All right. Other questions? Very good questions. Now, besides pores, you can have junctions that come together for adhesion and sealing. So here's your cells. And so a lot of times you want to really bring these cells together in a manner that they're very attached to each other. So they're involved in adhesion and sealing. And these generally work by having, I'll go over this in a second, what the specific complexes are. You've got a transmembrane protein. Here's the plasma membrane. Oops. These are transmembrane proteins with other intracellular molecules on the other side, inside the cell. So you've got transmembrane proteins, which are these guys, attached to each other from each cell supplying one. This is cell number 1 here. Cell number 2 over here. And you have intracellular kind of peripherally membrane-associated proteins forming a protein complex. These work through protein complexes. That's what's really important here. So, One of these, I might as well stay over here. One of these is called adherence junctions. These guys form belts of adhesion around the cell. These are very important. For example, you'll see in epithelial tissue. I'll give you a very more specific example. But they're also very important in, say, in cancer. When a cell metastasizes, it comes off a tumor, you have changes in adherence junctions, and then when it metastasizes and goes to another tissue, you have a different set of adherence junctions will form. And it kind of can regulate the ability of a cell to come off of a tumor mass, for example. Now, the transmembrane protein for adherence junctions are called cadherins. The intracellular protein is called catenins. Let me just make this a little bigger here. And there's different kinds of catenins. And what's important here is this all connects to the cytoskeleton. So the shape of a cell, because it's regulated by the cytoskeleton, also connects to these membrane complexes. And very important, therefore, in the architecture of a cell. Forms the shape, helps form a shape. Okay? Now there's another kind of adherence junction, adhesion, called desmosomes. These guys are spots of adhesion. Whereas adherence junction are belts, these are like little spots of adhesion. And here the transmembrane protein are also cadherins, but they're not quite exactly cadherins, so they're called cadherin-like molecules. Here's the plasma membrane. And on the other side of it are a variety of molecules. Desmoplakin is one notable example. Now, desmosomes are very important, for example, in keeping your skin cells together. And you can have defects in desmosomes. One of them, there's a blistering disease. It's a genetic disease that has a defect in desmosomes in the skin. So what happens is, because you're disrupted in skin cell-cell interactions, you have unusual movements of water in between the cells, and therefore you get these blistering effect. Okay, so something like, the manifestation of this in a disease state sometimes can be quite unusual in terms of these cell-cell interactions. Okay? Now there's another kind of junction that's involved in sealing. And the difference in sealing between adhesion, the analogy I like to use is if you have a jar, a glass jar, and you put a lid on it, you know those lids? They'll have a rubber kind of gasket around it. You guys all familiar with those, right? Take the gasket off, seal the jar, put water in it. What happens is you have contact between the lid and the jar, but you turn the jar upside down, what happens? It'll start leaking, right? So you can have adhesion, but you don't necessarily have sealing. But you put that gasket back in it, give it the adhesion, and the water stays in the jar. So you got a sealing effect. And you have different junctions that actually work together a lot of times, but it's called tight junctions and are involved in sealing. And we'll talk about how tight junctions and adherence junctions work together when we talk about epithelial tissue. So for the tight junction, the membrane proteins are called occludins or claudins. There's two different kinds. There's actually, I put this in plural because there's 24 different claudins. It's a fact you don't need to know for any reason, except if you study it. And on the intracellular side, it's called zonal occludin proteins. ZO1 is very important. And there's other proteins will form here. The point is, you get a protein complex creates the sealing. And as I mentioned, tight junctions and adherence junctions work together We'll talk about this for epithelial cells in a second, and we'll just get to that one second. Okay, any questions? The difference between sealing and adhesion, the difference between adherence junctions and desmosomes. Okay? Yes?
What structural feature of the tight junctions are different from like the adhesion junctions that make them sealing?
Okay, the question is, why is it that tight junctions seal and adherence junctions don't? Maybe that's, and why is it that tight junctions don't necessarily adhere? They do a little bit, and these guys are really important. What it amounts to is the nature of the function of these proteins. So these guys, these cadherins, when they come together, they're very tight. It's sort of like kind of like the super glue of molecules. They come together and they form a very tight interaction. Whereas these guys form a different kind of interaction that actually prevents movement of molecules. So, I mean, the short answer is there's the function of the although they're both transmembrane, they function differently. Okay, just the nature of their three-dimensional structure and how they interact with themselves and the cells themselves. Okay, but they work together, and we'll see this in a second. So where they work together is in epithelial cells for adherence. Now, just talking about adherence junctions and tight junctions. So now we're going to move to the different tissue types. There's only 4 different kinds of tissues. Tissue types, I should say. You have epithelial tissue. And the main functions is they're involved in protection. Epithelial tissue surrounds the organs. Okay? If you look around at your neighbor who's sitting next to you, you can see epithelial tissue, and that's your skin. And then everybody can easily perceive of skin as protecting yourself from the environment, right? It's epithelial tissue. Forms a barrier, as we talked about, between sealing between cells and adhesion. I'll put this up here in a second. Also heavily involved in secretion and absorption. Okay. So again. So epithelial tissue sits on a basement membrane when it surrounds organs, and just sitting there, like skin is a good example. And basement membrane is basically a mixture of proteins and complex oligosaccharides and so forth. And it forms a protection. As an example, UV light. If it wasn't for skin, you'd have all sorts of damage in your body. Now, not surprisingly, because epithelial cells are involved in protection, most cancers are epithelial derived. About 85% of cancers come from epithelial tissue, because cancer is a disease of stress and aging, biological stress. And where does the stress hit? Mostly it hits the epithelial cells. So therefore, cancers are mostly epithelial in origin. Okay, so it's not surprised that that would be the case. As I mentioned, epithelial cells, I'll give you some examples here in a second, are very important in regulating movement between cells and attachment to cells. So if here's 2 cell types, here's the tight junction, TJ for tight junction, AJ for adherence junction. They work as a complex. In fact, it's actually given a name. It's called an apical junctional complex. Okay, so this is a functioning unit. You can have adherence junctions without tight junctions, by the way. You can have adhesion without sealing, but you cannot have sealing without adhesion. So it's a prerequisite. So in terms, one of the things this does, again, these are belts. So both these guys are all around this. This board is 2-dimensional. Cells are 3-dimensional. Just envision this belt of a sealing and an adhesion all around these cells. So it sees two molecules, X and Y. The tight junction, combination of tight junction and adhesion junction, prevents X from getting in between the cells, but allows Y, molecule Y, to come through. So it's selectively permeable, but very selective. Very important. And you'll see this is extremely important in lots of physiological functions. In addition, the adherence junction and tight junction belts define different parts of the membrane. So this part of the membrane is the apical side of the membrane, and the other side, it's the basal side. So it defines different parts of the actual cell itself. And as a result, it defines different physiological functions. And I'll give you two examples of this. Now, during late pregnancy, we'll talk about this a little later, but talking about as you go from late pregnancy to lactation, there's a huge increase in tight junctions and adherence junctions. The apical junctional complex will form on mammary gland cells. The reason is, if here's a typical mammary duct, and it's heading out to the nipple, what you have on the side here are different cells, epithelial cells, or some of them, with very intense, intense is a bad word, very strong tight junctions. The reason is, during lactation, from the name, you're making milk. It's milk production. It's very, very important for the viability of the organism. And so what happens is, the milk proteins get secreted out the apical side of these cells. Now, and from the basal side, you have various hormones are telling the cells, hey, we're past pregnancy, we got lactation, we got to make the milk proteins. What you don't want is the milk to leak back in between the cells back into the blood of the mom. And you don't want the hormones leaking in between the cells through the milk into the baby, right? What prevents that? This apical junction complex, combination of tight junctions and adhesion complex, keeps these cells totally locked in terms of which side of the cell they're on. So the milk always comes through the duct, and the hormones always stay on the other side. Incredibly important. If it wasn't for tight junctions and adhesion junctions, this whole process would not function very well. As you can imagine, in breast cancer, one of the things that takes place is a disruption of these cell-cell interactions. And it's very common to see losses in ZO1 functionality or expression. Same with the cadherins. Cadherins can go very low, in fact, in some kinds of breast cancer cells for the disruption of this. Okay, is there any questions? Yes, in the back again. The question is, what happens when epithelial cells sloughs off? Intestines is a good example of that happening. You lose the, you need two cells to get a tight junction or adherence junction. The proteins will still be there, but you don't necessarily have an active tight junction or adhesion junction. Another example is metastasis in cancer. It breaks off the tumor mass, a cell will break off, goes somewhere else. It still has the proteins, but it doesn't form a junctional complex until it has in contact with another cell. So it takes two to tango, so to speak, for this. Okay? Other questions? Very good question. Yes? Very good question. Why are we so scared of cows treated with growth hormone? I am one, I'm not scared of that. Because for one thing, if you eat cows that are treated with growth hormone, in my opinion, your stomach digests it all anyway. No protein hormone is going to make it through the digestive tract. So I am not scared of that. But I have other things I worry about, like will Kentucky beat Kansas tonight? More important to me. Yes? Sorry about you guys that are Kansas fans, but I'm not. Oh, the apical basal. it's defined different ways. The easiest way to remember it is the basal side generally, but not always, faces the blood. So the hormones are coming in from the blood side. You don't need to know that this is basal and this is apical, by the way. You don't need to know that. But the point is, you've got two different sides of the cell which have different functions. That's the important thing. So that's why there's different functionality in each side of a cell. Incredibly important physiologically. Okay? Now, another example of this is your digestive system. So you've got these cells that are in the interior lining of the digestion, and these have very strong tight junctions and adherence junctions. The reason is you want these molecules to come into the cell itself and then out of the other side of the cell into the circulatory system. You don't want these nutrients to leak through in between the cells and therefore never make it to the circulatory system. So these have very strong tight junctions. And there's disease states where this is all messed up. There's a disease called multiple organ dysfunction. Basically, when all your systems are really falling apart in multiple organ dysfunction, one of the things that takes place is a complete disruption of your intestinal cell-cell interactions. As a result, people that have this and are really getting close to the end, they can't digest very well because they don't have very solid tight junctions in their intestines, and therefore they can't bring in the nutrients appropriately. So it's a very common kind of thing that takes place. Okay? So very important physiologically. Another one you don't need to know about, glaucoma, is involved in changes in pressure and changes in cell-cell interactions and tight junctions. So there's lots of disease states where that's involved. Okay? Questions on junctions? Okay. It's not just how a cell interacts with each other, it is also the shape of the cell plays a role in a lot of this. Shapes are important. And there's three basic shapes. One of them is squamous, which are flat, tile-like. They are also very leaky. And leaky is a good thing for some processes. And so we ask yourself, where is leaky good? Leaky is good in capillaries. So here's a capillary, which is made up of a bunch of epithelial cells. You have a bunch of cells, and here's the middle of the capillary. That's the blood going through the capillary. And this molecule wants to go in between the cell, and in between, let me just make this a little bigger here, wants to come out of the capillary into a tissue. Well, you don't want to give it a barrier. If it's an important nutrient, you want that molecule leaking right through. And the reason why capillaries are so good at what they do is they're good at being leaky. And these are made up of a single layer of epithelial cells, and a single layer is a simple squamous. And when we talk about the systems flyby, the entire thing works in part because of leakiness and is trying to establish an equilibrium between cells under different physiological states. And it's because these capillaries are leaky, allows this. Now you can also have squamous cells that are a little bit less leaky, but still a little leaky. And this is skin cells. You have what's called stratified. Squamous, which is skin cells. And if simple is one layer, what do you think stratified means? More than one. Very good. And so skin cells actually have 3 layers. And the reason why this is good is because of abrasion. So you fall, and I have 4 kids, and trust me, they're falling all the time. In fact, I carry Band-Aids with me at all times because of this. They come off, and they take off a bunch of cells, but you don't want to go all the way through and just bleed out. Okay, you want a little bit of protection. So stratified squamous cells allow some abrasion to take place without massive injury issues. So you have 3 layers of that. So it's stratified. Okay? And of course, there's all sorts of interesting desmosomes going on that's keeping the skin cells all attached to each other. Now I have another kind of shape called cuboidal. Oop. Which are kind of rounded or looks like cubes. And these are involved in specialized secretion. An example are endocrine cells that produce hormones. Not all of them are this, but a lot of them are. They make hormones. And these are cuboidal in shape. You've got another kind called columnar. Columnar are particularly good in making tight junctions and adherence junctions. And columnar look like columns. They have a very large cytoplasm for massive secretion and absorption of large amounts of material. And if I said to you there's lots of secretion and lots of absorption and or absorption, you think you can predict a place where you have columnar epithelial cells? In intestines, for example, right? Because you're taking in massive amounts of nutrients. And it's because the cytoplasm is big and it just happens to be columnar in shape. Okay? So shape is loosely, and I stress the word here, loosely correlated with functionality. Okay? Any questions on that? If, for example, capillaries were columnar cells with really good tight junctions, how would capillaries function? Good, bad, or indifferent? Bad, right. Because capillaries like to be leaky to function. If you have very strong tight junctions, you're not going to be leaky, and therefore they wouldn't function. So there is some connection between the shape and the functionality of the cells themselves. Okay? So just the way it turns out. There's something else you don't have to know about. There's something called pseudo-stratified. Okay, what does the word pseudo mean? Kind of a false looking, right? So pseudo-stratified is, you look in the microscope years ago and you say, oh yeah, that looks stratified. Looks like there's multiple layers. But when they actually tease it apart through a much better microscopy, it turns out to be a single layer that looked like multiple layers. And there's no obvious functionality for it other than mistakes by the original investigators to look at the shape of the cells. Your nasal passages happen to be pseudo-stratified in the way they kind of look in a low mag microscope. Okay? So names still stick, though. Now, connective tissue. Now we're done with epithelial. Let's go on to connective tissue. Connective tissue is by far the largest amount of cells, or some of the largest, and it's involved. The key thing here is, for connective tissue, here you have a cell, and out of the cell is a secretion called an extracellular matrix. And this can be lots of proteins, it can be complex oligosaccharides, et cetera, et cetera. The connective tissue is distinguished by the nature of the extracellular matrix. And it's a very broad cell type. Sometimes this extracellular matrix can be solid. So solid ones, for example, so here's the cell types. An osteoblast. And what's the matrix of an osteoblast? Bone. Obviously very solid. Unless, of course, some of you guys came back from skiing with broken bones and things like that. Sometimes it's not as tough as you think it is. I've broken many bones, so I know the feeling. You also have chondrocytes. Form cartilage. So these are solid. The extracellular matrix is solid. You can also have jelly-like extracellular matrix, sort of like right here. Okay? And that is adipocytes, or your fat. So it can be jelly-like. So you can have adipocytes. And the extracellular matrix is fat droplets. And you can also have liquid connective tissue. And that is your blood cells. Your blood is actually a connective tissue. And the actual extracellular matrix is the plasma of your blood. Okay? So it can be, think about this distinction. Plasma, fat, bone, cartilage are all connective tissue. That's a full spectrum of cell types, cell and tissue types. Now there's two other kinds of connective tissue that are worth mentioning. One is free connective, loose connective tissue. And the cells are fibroblasts. There's a lot of fibroblasts, and these guys make a bunch of fibers, protein fibers. They make collagen, to make collagenase fibers or collagen. They make elastic fibers and reticular. And what do these things do? Collagen, the collagenase fiber gives strength. Elastic is flexibility, and reticular is cross-linking. What this does is it holds all your organs together. It allows me to jump up and down like this, and my stomach is still remains where it is and not up in my throat or my brains or something. Okay? It keeps everything together. It also allows you to take your skin and push it out, and it falls back into place. It gives you flexibility. There is a disorder out there, I forget the name of it, where these people, you see these sometimes in old shows, where they can take their face and pull it up over their head, right? They have changes in the ratio of these loose connective tissue. I think they have more elastic fibers than collagenous fibers. The point is this holds everything together, okay? And the last kind of connective tissue is called the fibrous connective tissue. And these are ligaments and tendons. Ligaments hold bone to bone. Tendons hold bone to muscle. And if you go around looking at anybody that got into any sort of skiing issues, usually it's a ligament or a tendon, usually a tendon, or it could be one or the other that gets stretched, broken, or torn, because it holds together the bone with the muscles. In addition, people that are on performance-enhancing drugs, PEDs, as they say, they have in the news, a lot of times are very susceptible to tearing of ligaments and tendons, because the muscle gets so big that it's not designed. So if you build up a lot of muscle mass, your tendons and ligaments aren't designed, particularly tendons, are not designed to hold these together very well. So you're more susceptible to tearing and breaking. So that's one of the downsides of performance-enhancing drugs is tearing of these guys. Okay? Questions on connective tissue. It's all across the map. It's blood. It's fat. It's bones. It's tendons. Okay? It's almost like they decided, let's name muscle and nervous and epithelial tissue, and what's left is connective tissue. But the common thread is extracellular matrix. Questions on connective tissue? Okay, so let's talk briefly about nervous tissue. Now there's two basic cell types. There's a little more than two, but there's two general. One are neurons, and the other tissue cell type are not neurons. Okay? And I only say that because neurons are the ones that all the action is taking place, and the other cells are kind of like the support system. A very common non-neuron are glial cells. You'll see a little bit how these guys work. The glial cells, for example, can provide nutrients to the neurons, provide insulation to the neurons, as we'll talk about, actually affect the nerve impulse in terms of what goes on. Now, the nerve cell itself is a very straightforward structure, a little bit different than some of these other cell types. You have a cell body, and you have an input through a dendrite. It receives this electrical input. It goes down a long extrusion called an axon, where a lot of the action takes place. And then there's an ending to an effector cell or another neuron. And what goes across, this is the synapse. And what goes across here are neural, put this down in a second, are neurotransmitters. And these are molecules that bind. There can be peptides such as endorphins, small molecules, catecholamines like serotonin. They come across, they bind to receptors. This will become more clear-cut when we talk about hormones starting Wednesday. And they cause the effector cell to respond. Now, what goes down the axon is with something called a nerve impulse or an action potential. Let me just put this up over here, a little bigger. So we're going to take a little segment of the axon. In addition, the axon has got a myelin sheath around it. And let me just put this over here. So here's a blow-up of the axon. These are some cells. And this forms a myelin sheath, which is this part here. The myelin sheath is made up of glial-like cells. And these guys provide an insulation to the axon. And there's different names of glial cells. You don't need to know the names. Just something you don't need to know, but like in the peripheral nervous system, Schwann cells, you may have heard of that, form the myelin sheath. They are glial-like cells or derived from glial cells. Don't need to know specifics, except that it's a glial cells involved in this myelin sheath. But it also accelerates. The interesting thing to me is it accelerates the nerve impulse or the action potential. And what the action potential is this information that shoots down the axon. And it's a bunch of ions change. So here's this axon coming in there. So what happens is it gets an impulse or information from coming down through the dendrite. First thing that happens is these ion channels open, sodium ion channels open, and sodium comes in. And then what happens is that depolarizes the cell. What that means is not all that important at this point. And you stop, you close these channels, but then you open up potassium channels to bring the positive charge back out of the cell. In the meantime, further down the axon, more sodium channels open up. So you have this depolarization, so you bring in positive charge, and to counteract that, you bring out positive charge through potassium, but then the channels open up further down, and more sodium comes in. So you have the sodium in, potassium out, shoots down the axon. And this process is accelerated by the myelin sheath. It allows it to basically quickly skip through. Okay? It's kind of like when you're channel flipping on the TV and you want to go fast forward to a different part. It allows you to do that. It allows you to fast forward down the axon. And so the myelin sheath is involved in this acceleration of the signal. And that's why it's very important, obviously. Okay? Questions on nervous tissue? And we have like 2 minutes, so let me just mention we have muscles. We do have muscles. And just very, very briefly on muscle tissue. Again, muscles have all sorts of gap junctions between them. Muscle tissue can be skeletal, which is striated, which means under a microscope you can see these lines, which is part of the microfilaments, actin cytoskeleton. And these are skeletal is voluntary. That means I can take my chalk and write on the board. You've got cardiac. Let me just put this down here. You got cardiac tissue, cardiac muscle. Cardiac is also striated. It is branched. It is contractile. But it is involuntary. It'd be a tough day on the job if we had to sit there and say, okay, heartbeat, okay, heartbeat, okay, heartbeat, right? It's totally involuntary, although it is striated. And then you have smooth muscle. Let me just put it over here. You got smooth muscle, which is not striated. It's involuntary. How many of you had breakfast this morning? Very good. Well, you don't sit there and say, okay, muscle, digest, muscle, move it through, digest, muscle, digest. It's completely involuntary. What drives the food through your alimentary canal is smooth muscles, bringing it all the way down. So you can sit there and listen to a lecture, enjoy the morning, yet don't have to worry about movement and digestion during your digestion because of your smooth muscles. Okay? All right, so I'll see you Wednesday. Different day, but same time, same place.